Good morning and good afternoon to everyone. I'm Leslie Commey from Texas Tech University, and today I'll be presenting on the topic Insoluble Polyphenols Mediate A flavus resistance in peanut. This is my outline for the presentation. It starts with an introduction and ends with an acknowledgement. A brief background. Peanut, we know, is a leguminous crop which is grown for its edible seeds. 2/3 of its production is obtained from developing countries. And the two major constraints in these developing countries in peanut production is drought and disease incidence. Drought, on the other hand, has resulted in a very big problem, which is still existing to date. This problem is known as the aflatoxin problem. The aflatoxin problem starts from the drought condition providing the perfect favorable condition for A flavus to infect peanuts. These infected peanuts are then processed and also used, the hulms are also used to feed animals. The processed peanuts are then transformed to peanut cakes and peanut butter, which are consumed by humans, and we also consume animal products such as milk and beef. When peanuts are consumed in the form of peanut cake and peanut butter, it results in the transfer of an aflatoxin known as B1 and B2, whilst that of animals, when we consume animal products, we get aflatoxin M1 and M2. This causes several conditions in the human system, with conditions such as suppression of the human system, acute aflatoxicosis, liver cancer, and also stunted growth in humans. To alleviate this problem, there have been a strict restriction by EU, which has set a standard of 4 parts per billion for export of peanuts, and also in US, the regulation is about 20 parts per billion for exportation of peanut products from developing countries. This has resulted in a significant decline in peanuts being exported from developing countries and putting a lot of pressure on the peanut processing industry to produce peanut butter as well as to feed animals to meet humans' demand. There have been several progress which have been made to solve this problem. Today, I'll highlight a few. To begin with, there have been 3 different mechanisms that have been used to solve this problem. The first mechanism was to identify resistant lines and susceptible lines by using the technique known as in vitro seed colonization. This technique was able to identify about 3 resistance lines known as 55437. We have PI337394, and we also have J11. Another approach was using pre-harvest A flavus contamination level. They also identified J11 and some other germplasm lines. The last one was determined using the aflatoxin production level, and they also identified lines such as PI337394. However, there is no common germplasm which have been found to be resistant in all 3 backgrounds. But looking at this closely, we could see that the common denominator here was all these lines or all these resistance mechanisms were developed from either the peanut having only the seed coat as its protective layer or the hulms. So based on this, we decided to investigate the role of seed coat or the role of seed coat in regulation of the aflatoxin problem or in determining the A flavus resistance. So we hypothesized that seed coat plays a significant role in regulation of Aspergillus flavus and aflatoxin production. Based on this hypothesis, we settled on 2 objectives. The first objective was to elucidate the role of peanut seed coat and the effect of the extract on A flavus growth. And also the second one was to identify the specific soluble or insoluble compound that have inhibitory effect on A flavus growth and overall aflatoxin production. Our experiment was done in Lubbock, Texas. The experimental material we used was 55437 and TMV2, which have been reported to be resistant and susceptible. Our experimental design was done using a completely randomized design with 3 replications for each sample. To meet our first objective, we surface sterilized seed coats, peanuts with no seed coat and with seed coat using sodium hypochlorite solution. And then we inoculated them using aflatoxinogenic A flavus strain, and daily reading of the number of seed colonized was recorded. An alternative approach we used was we extracted soluble biochemicals from the seed coats that we carefully removed from 55437 and TMV2 using acetone, acetic acid, and water. Insoluble biochemicals were also extracted, and this was done after alkaline hydrolysis and acidification to break the glycosidic bonds which are formed by these insoluble biochemicals with other glucose compounds present in the seed coat. To remove the interference of acetone, which has been reported to have antifungal properties, we used the rotary evaporator to dry out all the acetone out of it and dissolved it in DMSO, which has a less antifungal property and to prevent it from interfering with our results. The soluble and insoluble biochemicals were then used for radial growth bioassay. Data was collected for the radial growth bioassay until the plate was completely full, and our control we used was a PDA media with DMSO. AUDPC value was also generated from the daily readings taken, and percentage of inhibition was also calculated. And the data was then subjected to ANOVA for ANOVA analysis using GenStat. And the means were separated using the Turkish HSD test. So from the first approach, which we used without seed coat and with seed coat, we realized that on the third day, when there was no seed coat, both resistant and susceptible line produced 100% seed colonization by A flavors. Whereas that of TMV2 on the third day with seed coat produced about 30% colonization, and 55437 produced just 10. This clearly indicates the significance of seed coat in regulating A flavors in peanuts. Our second approach, using the soluble and insoluble biochemical extract, our AUDPC data showed us that in terms of soluble biochemical extract, 55437 had a lower AUDPC value compared to the control and TMV2. And when you look at the inhibitory growth, we realized that 55437 also showed a percentage inhibition of about 43%, which was higher than that of and more significant than that of TMV2 and control. In terms of insoluble biochemicals, we realized that the AUDPC value was also trending in the same toe as the soluble biochemical. But however, comparing the two soluble and insoluble biochemical, we realized that the AUDPC value for 55437 was twice lesser for insoluble biochemical as compared to soluble biochemical, and this was same for TMV2. And when you look at the inhibitory effects too, you could see that for soluble seed coat biochemicals, the inhibition was about 72% for 55437 and was about 58% for TMV2. This clearly indicates that seed coat, the insoluble components of the seed coat, play a major role in regulating A flavors growth and serve as an extra barrier, which provides an extra barrier aside the cell wall barrier, which is formed by the seed coat. So therefore, it prevents the fungal hyphae of the A flavors from getting to the cotyledon, which provides the requisite nutrients for it to grow. In objective 2, we wanted to find out the compound which was present or identify the specific compound. So we did soluble and insoluble biochemical extraction, and we dried it in using the rotary evaporator. However, there have been reports of several compounds present in the seed coat, but then it has been reported to be used as antioxidants. Some of these compounds include phenolic acids and flavonoids. So we employed the SPE technique to be able to separate these compounds. And this is a picture of our SPE setup. We have a vacuum, and also what we call a vacuum manifold. A vacuum manifold. Then for the SPE procedure, in order to separate the two compounds which we are interested in, which is the phenolic acids and the flavonoids, we employed this technique. First, we wanted to separate the flavonoids from the phenolic acids. So we employed a C18 cartridge. This cartridge is just an organic cartridge, which is able to bind to the organic compounds present in the peanut seed coat. So this was the procedure we used. And then in order to remove the phenolic acids, we use a preconditioned NH2 cartridge. This preconditioned NH2 cartridge is having a cationic properties. And because the eluted compounds, which were the phenolic acids, were adjusted to a pH of about 7 to 7.4, the anionic component was able to bind to the cationic cartridges, and the other components, which were not phenolic acid, were eluted out. And the retained compounds were then fed to the Hplc for analysis. After using several extended standards to be able to determine the Hplc data, we found out that we found 8 phenolic compounds, phenolic acids present in peanut seed coat. Out of the 8, we used 7 for radial goat bioassay, and we did it at a concentration of 0.67, 0.33, 0.167, and 0.033. The goal is to identify the specific phenolic compound and at which concentration is it inhibiting A flavus growth. We also took daily readings and generated an AUDP seed value as well as the percentage inhibition as described early on. So this is the results from our SPE, and you could see that there, from my far left, we can see the crude fraction that was obtained from the NH2 cartridge. And we can see the flavonoid fraction and also the phenolic fraction. The crude was also fed to the Hplc to be able to see whether we are properly eluting only the phenolic acids, or we use that to serve as a control to optimize the SPE technique. And this is my Hplc results. And you can see we've identified 8 compounds and I've stated them as compound A to H based on the retention time that the soluble crude extract was able to produce as compared to the external standard. And this is the radial growth assay results we obtained after using the 7 specific phenolic acids. So for compound G, the first figure A shows compound G, and we can see that from my far left, we see that we have the control, then followed by 0.667, and we follow by 0.3 and 0.167. This was the same for figure B and figure C using compound F and E. However, figure D was used to show the clear distinction between the control, the compound G, compound F, and compound E at 0.667 mg per ml. This bar graph clearly gives us a big picture of the AUDPC value we recorded for each and every compound at a concentration of 0.6 to 0.003. We realized that the AUDPC value at 0.667.
Leslie, we're down to 3 more minutes, okay?
For compound G was higher as compared to all of them, as compared to the control and others. Then in terms of inhibitory effect, we realized that compound G at 0.67 and at 0.33 showed a higher inhibitory effect as compared to the control and other compounds. So to conclude this experiment, we accept our null hypothesis, which states that seed coat biochemicals, seed coat mediate A flavus resistance, and we realize that insoluble biochemicals play a more significant role in regulating A flavus resistance, A flavus growth, sorry. Then we also find out that phenolic acids also present in seed coat influence the rate of colonization in peanuts. The rate of colonization in peanuts by A flavus. So our future research will be geared towards identifying the flavonoid components and also testing its role in regulating A flavus growth, and possibly identifying or quantifying the phenolic acids and the flavonoid in 55437 and TMV2. And lastly, we'll be able to develop a biochemical marker which we could be able to use to predict A flavus resistant germplasm line, and we will encourage farmers to use that. I would like to acknowledge USAID, Feed the Future, for funding my research, as well as Peanut Innovation Lab. And I also want to thank Texas Tech and AgriLife for providing me with the necessary things to do my research, as well as ICRISAT. Thank you very much.
Thank you, Leslie. We have time for a couple questions. The first one comes and says, I didn't understand how you related the drought to aflatoxin contamination. Can you explain that further?
Thank you. So A flavors are seen to thrive more under drought conditions. So when there is drought, we realize that the percentage incidence of A flavors is high. So that's how I was able to correlate drought with A flavors contamination.
Okay, thank you. And second question is, what is the contribution of the phenolic acid compounds in the seed coat? How effective is that versus other factors such as irrigation and insect management, the other factors that are associated with it? Is it going to just be another component for aflatoxin management, or will it make a significant difference in terms of the breeding effort?
Thank you very much. It's a very good question. Actually, we are trying to, this is just a preliminary results we are getting. The goal is to try and look at other factors such as irrigation. So we are going to subject these results to irrigation and see whether there's going to be an effect of irrigation and other conditions on the phenolic composition and overall A flavors growth.
Okay, thank you. Thank you very much for participating, and thank you for your presentation.
Thank you very much.